Hi dear guys, welcome to the next lecture of cell cycle, and in this lecture we are going to see about anaphase and telophase. That means obviously before this lecture we have seen about the prophase and metaphase, and in metaphase already we have seen that how the chromosomes are arranged on the equator or equatorial plate, or it is also called as metaphasic plate. So in this lecture we will see that from the metaphase how the cell promotes or proceed towards the anaphase, what happens actually over there, because we better know what happens in the anaphase. The chromosomes which were arranged on the equator in the metaphase, these chromosomes they separate, okay, and after separation each sister chromatid is pulled towards the opposite end of the cell. So this thing is going to happen over here. That means how the two sister chromatids which were bind together in the metaphase, how these sister chromatids or how do they get broken down, this phenomenon we are going to see over here. So before that we should know there are certain mechanisms happens in our cell. The proteins that are unwanted or the proteins which are damaged, okay, these proteins are destroyed by the mechanisms of a cell. So unwanted or damaged proteins are destroyed by the cell itself, and the system that helps in this is called as proteasome protein complex. Okay, so proteasome protein complex or proteasome complex is the system which destroys the unwanted or damaged proteins, and in this way all the cells are in our body, they regulate the amount of protein that is synthesized in their cells. The normal amount of protein should be synthesized, because if there is excess of proteins are synthesized in the cell, then there is also problem, and if there is deficiency of protein, then also there is a problem. So the amount of protein should be maintained inside the cell, and this is maintained by proteasome protein complex. Okay, and the excess of proteins which are just broken down, okay, after the broken down of these proteins the peptides are formed, and from these peptides the new proteins are formed which requires for the cell. Okay, so in this slide we are going to see the proteasome protein complex. The names of the two proteins are given over here. First protein, that is the separase, and second protein, that is the securin. Okay, so what is the function of separase? They have given us separase is a protein that degrade cohesin ring that links two sister chromatids. Okay, so already we have seen in case of metaphase the two sister chromatids are attached to each other, and they are attached to each other by means of the protein, and that protein is called as cohesin. So cohesin is the protein that help in the attachment of the two sister chromatids together. Okay, and what separase protein is doing here, it is going to degrade the cohesin. Okay, it is going to degrade the cohesin, and if the cohesin get degrade, that means the two sister chromatids are going to separate, and after their separation these two sister chromatids are pulled towards the opposite ends by the spindle fibers. We will see that later on. And the next name of the protein is given, that is the securin. So securin protein particularly, it is found in the cytoplasm. Okay, it is found in the cytoplasm, and it binds to the separase. So this protein binds to the separase, that means securin binds to the separase, and it helps the separase to properly fold into the functional conformation. The meaning of this is that without the help of the securin, separase cannot do its work, or separase cannot perform its work. That means without the help of securin, separase cannot able to degrade cohesin. That means separase needs securin for the degradation of the cohesin. So when these events takes place, then and then only the onset of anaphase can takes place. Okay, for the onset of anaphase, for the starting of anaphase, this mechanism has to be happened. Okay. In the next slide, we will see the figure of the anaphase. Okay. In this figure, you can see what you can see. You can see the two poles are shown over here, the upper pole and the lower pole, upper end and the lower end. On both the ends or at both the ends, centrosomes are found. Centrosomes are present. Okay, the upper centrosome and the lower centrosome. And you can see in the figure, from those, that means from both the centrosomes, the different types of spindle fibers arises. You can see there are three types of spindle fibers arises from the centrosome. On the top side, you can see in the figure, first one is the astral microtubules or these are also called as astral spindle fibers. Okay, these are very small one. Number second, kinetochore spindle fibers, or these are also called as kinetochore microtubules. Okay, that means these kinetochore spindles or kinetochore microtubules, they binds to the kinetochore protein found on the centromere of a chromosome. In last lectures, you already seen this, that on the centromere, the kinetochore protein is found, kinetochore protein is attached, and to that kinetochore protein, the spindle fibers are going to bind. Okay, and after that only, after the separation of the two sister chromatids, these sister chromatids are pulled towards the opposite ends, that we can see in this anaphase. Okay, so kinetochore microtubules. And third type of spindle fibers, you can see polar spindle fibers or polar microtubules. So these are the three types of microtubules, or these are the three types of spindle fibers you can see in this anaphase. You can see the centrosomes are completely moves towards the two opposite poles or the two ends. Then later, the chromosomes or the sister chromatids that are shown over here, their centromeres are bounded with the kinetochore microtubules. Okay, so all these events you can see in this figure. In the next slide, you can see what events are going to take place in this anaphase. So in the first point, you can see before the anaphase, okay, that means before anaphase, we have seen chromosomes are arranged on the equator in the metaphase. We already have seen in the metaphase, chromosomes were arranged on the metaphasic plate. So in the metaphase, the two sister chromatids were attached to each other. We have seen in metaphase, both the sister chromatids were attached to each other, and the X shape of the chromosome can be seen over there. Okay, and that attachment was brought about by the protein called as cohesin. So just before some time I told, the two sister chromatids are connected to each other by the protein called as cohesin. And in this anaphase, both these sister chromatids must be separated from each other. So they are going to separate over here. As I told just now, protein securin and separase, they destroy the protein cohesin. Okay, just we have seen cohesin is the protein that helps to bind these two sister chromatids to each other. And if these two sister chromatids should separate, then the cohesin must be broken down or must be destroyed. So the destruction of this cohesin protein is brought about by the securin and separase, which are the components of the proteasome protein complex. Next point, the two sister chromatids separated from each other by this mechanism, and after this, the two sister chromatids are pulled towards their opposite poles by the spindle fibers. Just now we have seen in the figure, to these centromeres of the sister chromatids, the kinetochore spindle fibers are attached, which pulls these sister chromatids towards the opposite pole. Okay, and you can see that when the sister chromatids are pulled towards the opposite ends, then the direction of the arms of the sister chromatids is towards the metaphasic plate. Okay, and these sister chromatids, they appears like banana, you can see in the figure. And next point, polar spindle fibers and astral spindle fibers. Okay, we have seen that kinetochore spindle fibers, they bind to the centromere, but what is the function of the remaining two spindle fibers? the polar spindle fiber and the astral spindle fiber, they create the forces. Okay, they create the forces, and due to that forces, the cell becomes oval. So in the anaphase, you can see the shape of the cell is oval. And why the shape of the cell becomes oval? So due to this mechanism. Okay, two spindle fibers. In the next slide, we can see main events in the anaphase. Okay, what are the main events in the anaphase that you should remember for the examination point of view? Centromeres split and chromatids separate. Okay, we already have seen the centromeres get split, and how these centromeres get split, that also we have seen. The cohesin protein is broken down by the securin and the another protein which is called as the securin and separase. Okay, so both these proteins perform that function. And second point, chromatid moves towards the opposite poles, that also we have seen. So with this, the anaphase is finished. That means what is happened in the anaphase, finally the sister chromatids, they move towards the opposite poles. So the next phase we are going to see is the telophase. Okay, so in this slide you can see the figure of the telophase, and in this figure you can see how the telophases differ from that of the anaphase. In the anaphase, the sister chromatids, they move towards the opposite ends. Okay, and here you can see the two daughter cells can be seen over here. Okay, the events seen in figure, the two daughter cells, the upper daughter cell and the lower daughter cells, these are formed from a single parent cell. Appearance of nucleolus, you can see. Appearance of nuclear membrane, you can see. Along with this nuclear membrane and nucleolus, endoplasmic reticulum and other mitochondria, certain organs, they also reappear. You can see the shape of the chromosome. Okay, that is also changed. That means cell is receiving the characteristic features of the interphasic cell. That means when the cell was in the interphase, at that time the characteristic that cell has, those characteristics can be seen in the telophase. So we are going to see about events in telophase. So the things that we seen in the figure, we will discuss over here. First point, this telophase shows reverse feature that of the prophase. Okay, so this is first point. That means what happened in the prophase, opposite to the prophase events are seen over here, and near about similar to the interphase. Okay, that means what we have seen in the interphase, that we can see in this, or we can observe in this telophase, but what we have seen in the prophase, just reverse to that changes are seen in the telophase. Okay, in the prophase what we have seen, the nuclear envelope was going to disappear, the endoplasmic reticulum, mitochondria, they were going to disappear. So likewise those changes were seen in the prophase, but in this telophase we can see the nuclear membrane regenerates, endoplasmic reticulum, mitochondria, they again regained. Okay, so this phase shows reverse feature that of the prophase. Second point, chromosomes assembles at two poles. We can see in this figure chromosomes are assembled in the two poles. In figure we have seen chromosomes begin to uncoiled or decondensed. In prophase we have seen the chromosomes were started for the condensation. Okay, they were going to become thick, and here just reverse to that condensation, the decondensation of the chromosome can be seen, and hence it is told that chromosomes begins to uncoiled, okay, and decondensed. Next point, chromosomes assume to take the characteristics of interphase, that just I told right now. Next slide shows us that around each chromosome, the nuclear membrane starts to regenerate. That means the set of chromosomes that we can see in at both the ends. So surrounding to that set of chromosomes at both the ends, the nuclear membrane or the nuclear envelope starts to develop, starts to regenerate. Second point, Golgi bodies, ER and nucleolus, these cell organelles also reappears. They again can seen in this cell. Next point, you can see in the figure, in the middle the furrow or the cleavage can be seen. Okay, in the middle of cell, furrow or cleavage is seen, and this cleavage plays very important role in the separation of both these daughter cells. So we can see that this furrow in the middle, later on it will get deepens in the in the next phase, that is the cytokinesis, and then after becoming deepen, the two daughter cells completely divide in the cytokinesis. But before this, in this phase, we can only say that the furrow or the cleavage can be seen in the middle of the cell, and then later the two daughter cells can be seen before the cytokinesis. So in this way we have completed this telophase. Okay, and in next lecture we will see the cytokinesis. Thank you.